So I've chosen for my title today, Dioxin Debates on the Downstream Biological Effects of Agent Orange. There are lots of biological effects we could talk about today. I know with the students here who are from ECOS, there's a whole raft of discussions we could have just simply about environmental effects, and while I think that would be a lovely topic, I think I'm going to stick to kind of our focus on adverse human health effects and talk a little bit about what's going on there. So I have if I can get the next slide. I can't remember if I was a clicker or not. Okay, so this is how I wanted to organize our work for the evening. I think in order to really understand this problem, we have to go back in time, back to kind of the beginning and frame the climate that was revolving around science and society in the 1960s. So this is a topic that will obviously be very imminent in the middle to the end of this decade. And there's some really interesting interactions between society and science, specifically the chemistry world, that kind of frames a viewpoint that I think many of us in the room may not totally appreciate in terms of entering this discussion. So based on that introduction, we'll transition into a discussion of plant biology. So a little bit of that kind of non-animal nature, okay, but mostly from the perspective of the chemist in terms of trying to mimic what we find in nature, specifically with respect to auxins. Auxins were the compounds that were modeled to come up with the interesting wartime tactic that the Air Force termed Operation Ranch Hand, but basically consisted of applying defoliants throughout the countryside in Vietnam as a means of routing out the United States enemy, the Viet Cong. And so we want to talk about the development of that agent as a warfare tactic, and talk about what is left over from that. And this is where we'll start talking about what we do with individuals who maybe have been exposed to something inadvertently and manifest with a variety of symptoms, and what we as a scientific world and as a community need to bring to that discussion in terms of supporting and facilitating a better understanding of those things. So through our discussion of kind of aftermath of Vietnam War, we can talk a little bit about how health assessments arose, what kinds of challenges and limitations are faced by individuals who are trying to get a better idea of what's going on there. And then, of course, we have a time point of 2012, where we are right now, where we can take a look at this and see where we are. And what we'll build towards here is that this is still a conversation in flux. This is a conversation that is debated by a variety of sides and will be continued to be debated for quite a while. All right, so next slide, please. This is where we will start talking about the climate, the social climate. Okay, here we are in the 1950s, right after World War II. Everyone's coming home, new industries springing up, and the world of the chemist has really exploded. The chemists have learned how to mimic lots of things from the natural world and maybe even improve on them a little bit. And in doing so, they're pioneering really important synthetic reactions that give rise to new things altogether. After all, this is also a time in which we see chemistry supporting space missions. Missions to the moon. Department of Defense is always a very good kind of catalyst for innovation, and the chemical companies, many of them that explode during this time, Dow Chemical, DuPont Chemical, Monsanto, petroleum-based companies are investing really significantly in the production and manufacture of new products, especially the products that make life easier, right? Gone are the days of washing your laundry by hand and hanging it out on the line. We want everything fast, we want it easy, we want everything to look as if we are daisy fresh and walking around as if it took no effort to do anything. And so I think you can see that illustrated in some of these posters. They're all kind of associated with Dow Chemical, but over here we see these kids. Obviously that candy is much better than the candy that Mom made on the taffy puller and, earlier in the day. Cellophane makes it better, a chemical product. Mother's Little Helper, anything that makes Mom spend less time in the kitchen preparing those lovely meals, right? Any kind of synthetic packaging, any pre-packaging, this is the rage, okay? And it's so important that Time Magazine chooses to celebrate, I guess, DuPont's CEO Copeland. Here, I love this little image here where whatever's coming out of the distillation vat of the day is the new product of the day, right? Shoes, clothing, furniture. It's all making life better, it's making life easier, and this type of advertising is permeating its way through society, right? This is very core, isn't it? Right? Those of you who are here. Okay, so in fact, this is the slogan. It's been modified over time by DuPont, but better living through chemistry, right? Who'd have thunk? Who'd have thunk that this was going to be something that got people excited? But you can see there's kind of a cachet towards chemistry and the chemical world, and chemistry as a way of solving problems, making our life easier and better. So this is kind of going on in the background now, and those chemists are kind of raised up in status. I'm sure all of you know of someone who perhaps dabbled in this industry at this time, and it was a boom time. It was a time of freedom and exploration and novelty, and the scientists involved here would have been enraptured by that creative mode that was going on, okay? And they're unleashed to explore all sorts of different things and maybe come up with the next saran wrap, right? This is great, okay. So it's in this kind of period then that we also see biologists digging down and trying to understand things at the chemical level as well. And biologists in the plant world were particularly compelled by findings about a naturally occurring plant hormone class called auxins. So before everyone glazes over, right, you know your biology class in high school or maybe beyond, those of you who are in my class in the back seat, this is going to look very familiar. Okay, auxins are naturally occurring hormones. And basically when you talk about a hormone, it's a systemic factor that influences the global regulation of the plant. Okay? Auxins in plants, especially and particularly vascular plants that have height by virtue of their stems, produce auxins at their tips, at the very emergent buds of the plant. And at this time, the idea that you could actually identify and purify one of these chemicals out of a plant was very exciting. And that was compounded by the fact that they found that it had a really interesting function. So not only do you have an up-regulation or an uber production of auxin at the tip of the plants, it turns out that as soon as it's produced, it travels downward in the plant until it reaches the root system, which is where the targets for the hormone are that allow for the cells in the root to start growing. Okay, so auxin as a hormone acts a lot like hormones in our body to tell the body to do major things, to grow, to make more cells, to turn cells from one type into another type. That's called differentiation. Okay? So this was exciting. The plant world found that they had a great model in which to understand growth, like living material as it expands. And they were very excited. And of course, being able, again, to take cues from the chemistry world to purify and examine the properties of this molecule was extremely exciting. So on the next slide. The major class of auxin, there are a few, but the major class found in most vascular plants has this hairy name, indole-3 acetic acid. Looks like something that came out of DuPont, right? But actually, this is a natural product. And again, those of you who are coming here with a little bit of biology under your belt, you look closely, you start to see an interesting double ring structure here. Right? This is a kind of a canonical structure that's used in ATP. It's used in all of the nucleotides that build DNA. So you can imagine that there is some kind of common compound that plants use and decorate with extra doodads to make it do its thing. Okay? So this is derived from natural nutrients that these plants would have, and similar to ones that would be present in you and me. Okay, so this is indole 3-acetic acid. The shorthand term is IAA, but this is the auxin. This is the thing that's produced at the tip of the plant, and after it's produced, it's shunted down to the roots to trigger a growth response or a differentiation response. Okay? This is the biology, and believe me, the biologists who figured this out, this connection out, were just giddy with their findings. This made them very ecstatic. They would be happy to see what's happened since then. And I dared to show you this awful, ugly mechanism slide because this is a biology talk, and I want you to know it's definitely approachable. I think if you look at that, the first thing that jumps out at you is all those arrows pointing in. So you can see that it's important for the target cell to actually import auxin into the cellular milieu. Okay? We have a couple of transporter proteins. It gets in by this mechanism called endocytosis. But once it's inside, that's when it does its important work in terms of directing cell function. So you can see IAA starts to associate around this little yellow circle. This is the cell's nucleus. This is home to the genetic information of the cell. And it turns out that when IAA gets into the nucleus, it can couple with proteins that serve as auxin receptors. Okay? Fussy word for a protein that is waiting to catch on to some auxin to trigger the change of gene expression. Gene expression, what I mean here is binding to a bit of DNA, the chromosomes, causing the chromosomes to generate some RNA, which is an intermediate kind of message molecule, which then converts it into protein, and then it's the proteins that actually do the work of building the cells or changing them from one type to the other. Okay? So auxin does this. This is its functionality, and I wanted you to see a little bit about how far down we can drill so that you can understand that a molecule like this, in order to interact in this environment here, has to be specifically shaped and exquisitely tuned to these types of systems. And again, the biologists who figured this out, jumping up and down, we have the magic bullet, right? If we know how to grow bigger, better plants, imagine how rich we will be. Right? Okay, next slide. So the work went on in earnest with the biologists collaborating with the chemists who were already making life better to come up with synthetic compounds. Now, why do we need synthetic compounds? Well, it turns out that if you have a molecule that is a subtle regulator of growth, it's usually present in very, very small amounts. You don't want a lot of it on, you want it in low levels so that when something has to happen, you can boost it up and then take it down. So you can't extract a lot of auxin from a cell, and you might need to make it on your own in a laboratory where you can make it by the bucketful. Okay? So, and that's exactly what happened. And there's a gallery of synthetic auxins that are out there, probably 5 or 6 that I ran across in preparing this talk, but the 2 stars of the day are these 2. 2,4-dichlorophenoliacetic acid, 2,4,5-trichlorophenoxyacetic acid. They actually, even by name, sound very similar. But if you look at the compounds, right, it's not too hard to see. They're pretty much the same thing, except for one has 3 chlorines and one has 2. But they both act like auxins. If you deliver them to a plant system in reasonable doses, they will also exert pro-growth and pro-differentiation effects. In fact, T4D here, this is something that's sold in every Home Depot across the country. It's sold in a little powder. You rub it on the end of your leaf or a stem that you've picked out of the garden, and it encourages the growth of a root system. Okay? So this is a synthetic form of a plant hormone that actually can help encourage an appropriate response in plants. Turns out that as they're making it by the bucketful, they're saying, well, how much can we add to make, the Jolly Green Giant's, tree that he's going to hang in? And so they kept throwing more and more and more on, only to find that at a certain level, auxins are toxic. And they're toxic not because they cause plants to shrivel up and die. And I say that because in the video that some of you might see tomorrow night, there's a lot of footage of just kind of dried out, crumbling vegetation. No, These synthetic auxins, when applied at high doses, actually inhibit growth. So the idea is that if you can swamp a system, a garden or a crop system with excessive amounts of auxins, then you'll get stunted growth of the organisms. Now, the type of plants that are affected by overactive synthetic auxins are a particular class of vascular plants. Those of you who are in biology have been talking about monocots and eudicots. Monocots are the grasses, kind of thin blades with limited vein systems. Eudicots have broad leaves with very complex vascular systems. These work on the broadleaf plants. So when you are choosing an herbicide to use in your garden, on your lawn, right? Here we are in Atlanta with our perfect lawns. You can imagine life could be better if you could apply a compound that got rid of the broadleaf dandelions without, sweating a drop, right? Life could be better. And that was the idea. That was the idea. If we have these in hand, life will be better because we can get rid of those nasty weeds, and we will do this in a way that is consistent with what the chemists teach us. Okay? So these are these synthetic auxins. This is one of those ideas that in theory is fabulous, right? I don't want to sweat. I would love to look like the lady in that propaganda cartoon we just saw, all fresh and ready to engage in snappy repartee. But the military actually had a different idea, right? They saw this as another opportunity. So the next slide. And this is where we get to talking about an Operation Ranch Hand, which was an Air Force-led initiative during the middle of the Vietnam War between 1962 and 1971. The reason why this was on their radar, and actually had been for a while, and I'll get to that in a second, the reason why this was on their radar is that their enemy was very elusive and was often shielded by very dense forestry and jungle land in South Vietnam, where they were embedded amongst the rural peasants in the more forested area. So at one level, being able to apply in a broad sense, very liberally, defoliants that consisted of some of these herbicides like 2,4-D and the 2,4,5-T would be to actually remove some of that foliage cover. And that would help reveal supply lines, right, that were covered over with leaves and trees. It would also perhaps reveal the site of bases, right? Tactical places where the Viet Cong was actually congregating and planning their next move. But of course we know that this war was also fought in a very guerrilla style. There were no big phalanxes. There were lots of little individualized altercations, and in many cases the enemy was living side by side with the actual peasants. And so another idea here was to actually also use these defoliants to target crops, right? So the idea is if you can damage the crops of a strategically localized population, you would encourage the rural peasants who might be supporting the guerrillas to actually migrate towards the cities where they might get a little bit more support. And so the idea was to also strategically identify sites where support for the Viet Cong might be also limited. Okay, so again, up in the corner there, we see a picture of 3 planes that are spreading and spraying the area. A couple more pictures here to give you an idea of the density of some of the forested areas. But they also would want to use these same compounds to fortify their own bases and make sure that there was that the borders were clear so they can monitor their own bases. Next slide. A couple more pictures. Initially, this was a really small program. They targeted the Air Force. The Air Force had a number of intermediate-sized transport planes that could be outfitted with the materials that they would need to spray. Initially, there were 6, and they weren't used very often at all. But eventually, that program expanded, and I'm going to show you a graphic in just a few slides to kind of give you an idea of how much it ramped up. But at its heyday, there were 25 planes that were being run by the Air Force that were making regular missions to spray in the area. And of course, down here, I wanted to include this. Obviously, helicopters could be used, and there were people on the ground who were also spraying this stuff out of cans in the local areas as well. So in all of these cases, we're looking at certainly an operation that required human intervention, but it was not as difficult as, let's say, going in and manually deforesting areas. So this was kind of a remote tactic, if you will, not too far away from some of the tactics that we talk about in terms of the wars in the Middle East right now. Hands off, not too many people get affected if we can kind of do these larger scale interventions. Okay? The other thing, the global term that's used for this is, just slipped my mind, area denial. Right? So in military speak, the tactic here was an operation that would deny certain areas to the enemy. All right, next slide. Okay, here are, so this is where we get to talk about what they sprayed. There were a number of formulations put together that are described generically as the herbicide rainbow. There were various combinations of chemicals, including the two that we were already introduced to, the 2,4-D and the 2,4,5-T. In addition to that, an arsenic compound called cacodylic acid was used, and arsenic, that also has kind of a drying effect. So when you look at some of the pictures where there's kind of dried leaves that are falling off of things, that's probably having to do with the cacodylic acid. Another component, pichlorum, not in the same class, the auxin classes as we were talking about before, but also targeting broadleaf vascular plants. Okay? And kind of put together in various combinations. The one that was used most often was known as Agent Orange. And over here you can see a very blurry photo, and I apologize for that, but you can see a collection of cans in which Agent Orange has been stored and is being prepared for delivery. And it was common practice to actually mix the herbicides with jet fuel, and that jet fuel was actually what was distributed. And so in the video that some of you might see tomorrow night, you'll hear people talking about how it was sticky and slimy if they felt it fall on them. Okay, so Agent Orange would have an orange band around it, Agent Blue would have a blue band around it, Agent White had a white, and so on, as a way of identifying them. Here we see on the top of one of those canisters a little bit of detail about its components. This Agent Orange was purported to be 50% 2,4-D and 50% 2,4,5-T. 50-50. Auxin, synthetic auxins meant to simply stunt the growth of vascular plants. All right. So over here on your right, then we see a diagram kind of talking about how much of this stuff was sprayed. 19 million gallons of agent whatever. 11 of those 19 were Agent Orange. Okay, so 19 million gallons. Olympic-sized pool times 40. Okay, so that's kind of the quantity that we're talking about that was handled by the veterans of Operation Ranch Hand and distributed. And I'm going to show you a pretty blurry map in a second, but a map that kind of talks about where the spraying happened. It happened over 6 million acres, which again, an analogy, 2 states of Connecticut. Okay, but spread out tactically across the country and actually eventually into Laos as well. So what we see here kind of is a graph that illustrates the point I was making before. Very early on, not much Agent Orange, Operation Ranch Hand was happening. Okay? In fact, President Kennedy, who was handed this as an option, was really resistant to it and did not want Americans to be involved in this type of thing. Eventually, obviously, someone had a better idea. In fact, and again, I am not a history buff with respect to the Vietnam War, but what I understand is that the Gulf of Tonkin incident, in which American ships were targeted in an attack by our Vietnamese enemies, kind of escalated the inclusion of Operation Ranch Hand more aggressively across the country. So this is where we see in 1966 huge amounts of, or relatively large amounts of material being sprayed, and the majority of it being Agent Orange. Let's see, is there anything else? Okay. The other thing I guess I just would mention, when the formulations were made, they really wanted to make sure that they hammered the leaves, and so the average concentration in these barrels, although you can imagine it probably ranged quite a bit depending on who was handling it and how they applied it, was about 13 times higher than what the garden label would have told you to use it on your dandelions in your lawn. Okay, so they hadn't done this before, so obviously they were trying to make it more likely to work. Okay, next slide. Again, so I apologize for the quality of the map here, but I did want you to have an idea of how far and wide they sprayed. Clearly, Vietnam here is cut up into four tactical zones, and they vary with respect to how intense the spraying was. When you see a region like this that's really red, these are the kind of the rural areas that were home to a lot of the Viet Cong, and sometimes in these areas, one place would be sprayed 2, 3, or 4 times. Okay. So kind of varies, looking specifically for roads and when it seemed relevant to actually target crops in the field. All right, so next slide. So this is kind of a gutsy move, I think. It's kind of brash, but it's definitely consistent with this chemicals makes our life better. Chemical makes it easier to fight the war. And it's consistent with that. In theory, there was a lot of scientific backing here, and probably made individuals feel pretty confident that this was going to be not so invasive as actually dropping bombs. Okay, because the target should have been just the plants, the auxin targets. So here's a shot of JFK's cabinet room early on. This was not an easy decision for implementation. In fact, at this time in the 60s, we're coming off of a period where we're really trying to figure out, do we want to be involved in any type of biological or chemical warfare? There are discussions about whether this is akin to chemical warfare, and of course those who would defend it would say this is not directed against human beings, this is directed against plants, so it's not the same thing. There were certain politicians associated with the administrations who pushed back and said this is equivalent to imperialistic barbarism. We don't care that it's just directed at plants, but the act of going and swarming and doing this kind of aerial delivery of this agent that could potentially affect the livelihood of the residents in the area, that wasn't consistent with what it was to be American. I was also kind of struck to find out that the scientists were pretty active here. Initially, the Federation of American Scientists had about 5 of them in the early 60s write letters to the science advisors of the president saying that this was not an appropriate use of the technology, and that sentiment built over time to the point in the late 60s where 5,000 scientists signed a petition against the use of herbicides in warfare. 17 of them were Nobel laureates, and 129 of them were members of the National Academy of Science. And I want to spend a little bit of time talking about the National Academy of Sciences because they're going to come up at the very end. Everyone who works in the scientific world engages in their career with the hope that one day that they will be elected to the National Academy. The National Academy is an elite group of scientists who basically is used as the think tank, the scientific think tank for our country. And they are well regarded. You are only nominated and gain access to the National Academy of Science if you have had a long and effective career, if you have shifted paradigms, if you have pushed your field, and if you have been a leader, both in the laboratory as well as in the community. So, so getting these kinds of comments from the scientific community, I think, was a little bit of a pushback to this chemistry will make life better, right? Maybe it will, maybe it won't. Let's make sure that we address it scientifically before we buy into it. So there was all that kind of pushback, but obviously not enough to abort mission early on. It happened later in 1971, and partly because they had already engaged in this idea for a while. They had played with formulations of herbicides, maybe to use at the end of World War II in the Pacific Rim, and they also had seen it work well for Britain, who suppressed insurgents in the Malay Peninsula, and they thought that this was a reasonable way to go. Okay, so it's kind of like this is this tension, right? Who's got the best idea here, and who already, who's taking ownership for this kind of decision? And it's you can't really make that call here. And we certainly don't have the faces of the individuals who were necessarily pushing it as hard as some other ones. Okay, so there are precedents for the use of herbicides, but serious objections to herbicides. And so it was probably not any surprise that after the Vietnam War, there was a lot of criticism. Anecdotal commentary coming from Vietnam veterans coming home after having been exposed to Agent Orange, Agent Blue, Agent White, and remarking that they didn't feel well. Longer term, remarking that they were having problems having children, and if they did conceive children, that their children were somehow not quite right. And at the same time, we're hearing those from the American soldiers. Other anecdotal stories are slipping out of Vietnam as well, and of course, these kinds of structures that we have, sculptures that we have around us right now, is thematic to what we're talking about here. Here is a picture from a hospital in Vietnam where you can see a collection of children, many of whom suffer from physical malformities, developmental disorders, some type of intervention during development that prevented them from growing fingers properly, growing arms properly. Many of these individuals were born to very poor peasants who don't have the means to support them. Not only are they physically impaired, but many of them have neurological issues, suffer from mental retardation, and so they do exist as kind of the culmination of some of the problems that the local Vietnamese people experienced in the aftermath of the Vietnam War. Okay? So because of these stories about seeing stuff falling from the sky and feeling it on your skin, those anecdotal stories made connections with what caused it already, which, it's the normal thing to do. When something happens, you want to know what caused it. So that urge to find cause and effect is something that is intrinsic to us and right front and center when people are suffering tragically from these family difficulties. Okay, so lots of anecdotal stories coming in, addressing politicians, addressing the president. Something must be done. And along this time, as the rampage kind of brewed, another finding came to light. So on the next slide, what was discovered, and this was discovered at the beginning of the war, actually, and there's stories about how it was brought out publicly. But in that same synthesis that gives rise to 2,4-D and 2,4,5-T was another compound that was a byproduct of the synthesis called 2,3,7,8 tetrachloro-dibenzodioxin, the big D word, dioxin. So this was not an intentional product. This was a product of incomplete synthesis that has to do with the fussiness of temperatures that need to be in the synthesis mix during the procedure. This is it's called the synthetic auxin congener because it was one of these byproducts. But right away, once an impurity was found existing in those vats of Agent Orange, those chemists and biologists went to work trying to find out what that thing did. We knew that the other things were kind of mimicking auxins, but what was this doing? So the initial round of studies on TCDD was simply to take it in its pure form and feed it to laboratory animals and find out what happens. So the first round of these studies was to feed it to guinea pigs, and they recorded that the dosage of TCDD that was required to kill the guinea pigs was the lowest lethal dose that they had ever seen of any man-made compound, and thus led to this quote down here citing TCDD as the most toxic man-made substance known. Toxic due to its lethality. Okay? The dose that needs to be fed to the hamsters in this case was so low, on the order of, I have it written down, I think it was 0.5 micrograms of TCDD per kilogram of guinea pig. So speck of dust, right? So this is what got everybody kind of worked up. The anecdotal stories, some work in the lab as a first pass showing that this could be a problem, and everyone got very, very upset and clamoring for this cause and effect relationship. So pretty soon afterwards, conversations back at headquarters for the Veterans Administration insisted that this be looked into. Early studies in the 70s and 90s were implemented, first just kind of an overall survey of immediate health effects in veterans, and also looking at the ecological problems in Vietnam. Basically, after a 3-year kind of investigation there were no significant problems seen in any Vietnam veterans in terms of health problems, and they also decided that any damage done to the ecosystem there would be replenished, although they did cite that the mangrove trees in southernmost Vietnam probably wouldn't do so well, that they would have to have a very active reseeding project. But they said other than that, everything was pretty peachy keen. But obviously, more anecdotal stories came out, people describing conditions, many of which now are on the verge of kind of being chronic with long latency periods, right? So when you're exposed to something, kind of like if we're thinking about an infectious agent, you have an acute response. It happens immediately. You go eat something bad at Taco Bell, that night you know about it, right? But for these longer latency diseases that we're talking about, neural dysfunctions, cancers, diabetes, all sorts of things like that, these are decades-long latency periods. And so people continue to be cropping up saying something's wrong, and I think it's because of my activities in Vietnam. So in 1979, another study was assembled, this time a cohort study, which is important because it focused specifically on the veterans associated with the activities of Operation Ranch Hand. Okay, so specifically looking at those individuals who are most intimately associated with these compounds and asking specifically about them, what kinds of things are happening to them. Right? Because if there's going to be a relationship between exposure and effect, those guys would be the target. And again, in 1979, despite extensive examination, no significant health effects were reported. Okay, so this is also gloomy and doomy. There are people who are really concerned about their health and their families and their livelihoods. And enough of a public clamor is made to urge Congress, finally, in 1991, to commit to the study of the health effects of dioxin over the long term. And I'm not listing these to suggest that these are the only things that have been done. I'm giving examples, and then I want us to focus on the Agent Orange Act here. A congressionally mandated project tasking our friends in the National Academy of Sciences to visit this problem biannually. And their task was to make sure every 2 years that they would scour the literature that's being published in the academic laboratories, the government laboratories, the industry laboratories, to find out any information they can about health effects that are associated with dioxin exposure, Agent Orange exposure, cacodyllic acid exposure, perchlorum exposure, anything that was in those agent cans. And so the first report was in 1994 and has been going on ever since then. And actually, we're going to finish with some of their most recent results. But before we get to that, I want to give you some ideas about how these studies are done. You know, with the CDC right next door, obviously some of you may be already well versed in these approaches, but I think it's worth citing that from a scientific perspective, and again, this is my goal, is to make sure we're looking from a variety of perspectives. From a scientific perspective, the approach to making a link between cause and effect is not very satisfying because the tools that we have to approach human disease are very blunt. They do not slice through to the absolute answer. And it's very frustrating. And individuals who are not familiar with what has to go into these studies can experience a lot of frustration because it seems so obvious. But yet, in order for us to demonstrate scientifically an association, it has to subscribe to certain rigors. And try as they may, scientists working with humans, and those of you who are biology like me know, humans are terrible model organisms, right? You have to ask them before you do anything. You can't feed them anything that's going to make them sick. So you get a lot of information based on interviews and narratives. And sometimes you can stick them and collect some blood. But even then, you know, that's maybe a little bit too invasive. So next slide. Okay. So one of the first things that was done in terms of understanding this TCDD compound that potentially has these awful effects is to drill back down into those chemicals and molecules. We saw this looks like Auxins, doesn't it? Different cell, right? We're in an animal cell now, but the same thing. What people have found is that when cells, or cells in animals, are exposed to high-level TCDD, there's actually a target, and the target is in your liver. Liver cells produce a lot of a protein called aryl hydrol receptor, or AHR. It does so in an attempt to clear your system of toxins. So here we have TCDD, it's coming in your body, it looks a little bit foreign. Our liver cell is actually trying to grab it and do something with it. And in the case of this particular cell, once the dioxin or whatever compound it associates with enters the cell, this is now competent to again move into the nucleus, and in the nucleus it also can change gene expression. This time we're not necessarily changing growth or differentiation. This time we're turning on genes, and these are genes that are involved in detoxifying our cells. So our cells are trying their best to get rid of this stuff, but it seems that they still have changes in gene expression that are above the norm or different than what's normal. They have been shown to undergo changes in differentiation. These are two big ticket items for biologists. They show aberrant behaviors. So you have a plate of cells, you put in some TCDD, and they don't act quite right. But it's a very minimalistic model, right? In our bodies, our cells have all sorts of barriers and circuitry that prevent kind of the environment from interacting directly with our cells. So although these studies are helpful, they don't necessarily tell us about the complexity of interaction system-wide. Okay, so next slide. So that's when epidemiologists have to resort to using the actual animals or humans as models. We'll talk about animals in just a second, but a lot of the life of an epidemiologist, including those that were tasked at exploring the causal association between various diseases and Agent Orange exposure, had to deal with these problems, these kind of impediments. So the two biggies when you are approaching something like this is to make sure that you are aware of any potential bias that you might bring into your experiment, and also to limit your confounders. Very fussy words, but basically all of the things that are happening here are streamlining the experiment so that you're always going to compare apples to apples. If at the base everything is the same, but one thing is different, then you can make a conclusion about what might be causing that. So bias here referring to a systematic distortion of results in a certain direction caused by the failure to account for some influencing factor. So Bias. We all kind of know what bias is, but it could happen at the level of the experimenter. It could happen because some people go to the doctor to get examined and some people don't. So your level of detection is maybe skewed. Recall bias. Again, when we're talking about these long latency diseases, 5 years after the fact, 10 years after the fact, 15, your memory changes, and sometimes that leads to misleading information in terms of demonstrating cause and effect. Selection bias. Those people who volunteer for Operation Ranch Hand Health Study, they're volunteering. And maybe we're missing a cohort that is worse off or not bad at all, and they don't volunteer. So these are things that have to be dealt with. They're part of experimental design. They don't sit well with individuals who feel bad right now and want an answer. They don't want to be told about bias, and understandably so. But in order to conform to scientific practice, the playing field has to be leveled somehow so that you're comparing apples to apples and that you can drill down to look for specific changes. Confounders, this is also going to be relevant, especially for a military population. So veterans involved in Operation Ranch Hand, yes, they had a common exposure, perhaps. But after the war, they went off and lived here and there and everywhere. Lots of different behaviors. Some smoked, some didn't. Some drank, some didn't. Some did drugs, some didn't. Okay? And those are additional chemicals in the system that may influence somehow the results that are being seen. So these are all kind of problematic. Okay. Epidemiologists, ever since the 1950s, when an association was made between tobacco smoke and lung cancer, have been trying to come up with guidelines to use, to hang their hat on, to demonstrate cause and effect. And it's very hard. This is a list from Sir Austin Bradford Hill, published in the 1950s, listing 8 different factors that must be true in order to demonstrate causality. That's a lot of hoops to get through, right? So there has to be a pretty decent risk of association between the exposure and the disease. Some of these have to do with the biology, is what you're seeing making sense with what you know about biology? Has it been seen in other people in another situation? Is there a proper temporal exposure to time of disease organization? And these things kind of have to be elaborated on. Some of them are easier, some of them are more difficult to actually assess. Next slide. And then, of course, it's the type of study that you perform is going to influence your outcome as well. There are 2 different types of studies that are done in epidemiological research, retrospective and prospective. Prospective is if a study starts here after some particular exposure might have happened and then goes forward from there. Retrospective deals with a start site that is beyond either the exposure or the emergence of the disease. And of course, when we're talking about those cohort studies, those early cohort studies, we are looking at a situation here where there is a common or known exposure, and they're tracking them later on to see if there are any commonalities with respect to their health outcome later on. So there are ways to go about this, but none of them allows you to kind of start from the beginning, take a control group that doesn't get treated, another group that gets treated, and then follow them both in parallel, which is, that is the golden approach in the scientific world. But they've outfitted themselves with some tools, however blunt they are, to try and get at this. Okay. Obviously, another component here is animal testing. If you take a TCDD, we already talked about how that was kind of fed to guinea pigs, leading to a really toxic dose. A really interesting story here is that subsequent animals were tested, and it was found that the lethal dose was 8,000 times higher than what was seen in a guinea pig. So the first test animal happened to be extremely sensitive, but our generic garden variety lab rat, which we use to justify pretty much everything that we do in the medical world, tolerates it at 8,000 times more concentrated levels, which is kind of interesting and historically is problematic in terms of which model do we hang our hat on there. It adds yet another problem. But obviously, certainly the pros of using animals is that you have experimental control, you can test increased dose and whether that leads to increased health outcomes. And we know that there are correlations due to evolutionary similarities between animals like rodents and humans. But still, we have some problems. There are differences, and they exist not only with respect to physiology, but also with respect to pharmacology studies. And I'm going to skip that one so that we can get on with this. Go ahead. The other problem, or the other thing that is very helpful to epidemiologists is a tragedy. This is kind of a two-pronged problem here. When scientists are trying to come up with reasons or an association between an exposure and a health outcome, they certainly don't plan on these types of mishaps. But when something comes up in which there is a blatant timed exposure that recapitulates what they're trying to ask, sometimes it can be helpful. And so in 1976, there was a chemical facility that was producing 2,4-D and 2,4,5-T and having the same problem producing congeners. And on one particular day, this chemical plant exploded, releasing a huge cloud of dioxin into the immediate and outlying area. So 11 local communities in northern Italy were exposed, children, men, women, elderly people, and although, pretty awful explosion, very terrifying, lots of alarm, especially given the uproar about dioxins, but historically, as we go forward, and this is relevant to the Agent Orange story, this is a large-scale human exposure to dioxin that is also being followed up and comparing cohorts to see if there are any commonalities. One of the things that you'll see on the last slide I'm going to show you is recurrently the acute effect of dioxin exposure is chloracne. So exposure to these compounds results in a pimpling of the skin. It is reversible, so it goes away after a couple of weeks. Obviously pretty traumatic here, but this is the #1 thing that is associated with dioxin exposure is chloracne, which is not a long-term system. Okay, next slide. Long-term effect. Okay, so this is the job then of the National Academy of Sciences, and in their purview, approaching this particular question, they have produced reports every 2 years, and they are sponsored by the Institute of Medicine, or the IOM, and in their report they summarize what the state of the project is. Have we identified any other diseases that can be associated with the exposure to dioxin? And in fact, their task was only to demonstrate association, not cause and effect. So over the years, as they have collected these data and reflected on what's going on, this list down here is are the only diseases that they have reported back to the Veterans Administration as being associated with exposure to dioxins. Okay? And this would include data from the Seveso incident, other occupational exposures, to try and zero down on what's going on there. So what we see here are a variety of very specialized cancers, lymphatic cancers, soft tissue sarcomas, as well as leukemia. So not solid tumors, but rather circulation-based tumors. So this is from the 2010 report. They have lists of other ones that are suspected, various intermediates, but these are the ones that the Veterans Administration, next slide, draws from in terms of making their recommendations as to who gets compensated long-term. Okay? Which, again, as you look at this, it certainly looks like very heavy-handed with respect to government, but the whole system is based on kind of feeding back onto the science and really trying to say what exactly it is. I don't think that scientists are really happy with the current list of associations. We, as scientists, kind of talk about, well, we don't know a lot of information that might be relevant here, and that kind of clouds the issue. But this currently is, this came off of the VA's website yesterday in terms of those conditions that can be compensated in some way, shape, or form. I'm positive it's not enough for those who are suffering from these problems, but this is kind of the state of the art. Next slide. So I certainly don't find this as a conclusion. It's not satisfying. There are many things that we don't know. It's kind of the nature of learning about these things. The debate goes on both sides. This is one of those examples where a very emotional subject confronts science in a way that maybe is not so flattering to particularly the science world. And I'm reminded of, on the next slide, a quote that I draw from in my core class on science. It's a quote by Stephen Jay Gould, who is deceased about 5 years ago, but a great paleo scientist and a leader in the field of evolution. And he described the situation that we are dealt here with respect to dioxin exposures. He would describe it as being an example of a great asymmetry. An asymmetry in which technical progress gone ethically and socially awry has come to bear. And I love the last statement from his paper that talks about the great asymmetry, and I'm just gonna read it off the slide. He, as a scientist, kind of talking to the rest of his scientific peers, says, "Wouldn't we achieve a crowning glory for our unique intellect if we joined the power of science with our unrealized potential for decency to rein in the great asymmetry and to build a just world that finally tempted us to exclaim with joy, 'Stay then, for you are so beautiful.'" And I think really this is where we are with this discussion. We are looking at a great asymmetry, we are trying to learn from it, and perhaps there are a few who are listening who are preventing that kind of problem from happening again. And that's, I think that's where I'll leave it tonight. So if you have any other questions, I'm happy to entertain them. Yeah, Marcus.
So, with our advancements in scientific knowledge now and sort of this system where you essentially will collaborate with scientists abroad, is there a growing knowledge of side effects of Agent Orange?
That's a good question, and one that I forgot to address while I was going through my slides. In those same reports that come from the Institute of Medicine, in the recommendations, one of the things that is addressed there is this asymmetry between attention to this problem and the health effects happening in the United States versus, let's say, in Vietnam. And we don't necessarily appreciate it every day, but we have institutions and structure built in here with things like the EPA and the CDC and other health interest group that keep us focused there and contribute money to exploration in this regard. In Vietnam, they don't have that kind of infrastructure, and although there are tendrils of hope that collaborations can happen so that scientists in Vietnam can interact and share their information here. The scientific platform, the structure, the rigor of the practice of science in the Western world is not similar to what's happening in other countries. And so although there may have been studies, perhaps in Vietnam, looking at the same problem, they don't subscribe to Sir Bradford Hill's proof of causation. They don't maybe practice as many controls that allow us to really dig and understand those differences. But my understanding is that there are, in progress, many kind of coalitions that are trying to bridge that gap and get more information from those who live in Vietnam who have not been tested, right, and who have not been part of larger health effects studies. Yeah. Oh,
well, I just have a great trouble, it seems to me, hearing that we're quite cavalier in inventing these things and using them. and much more demanding and rigorous on saying, prove that we did some harm. It pisses me off, I'm sorry. It just makes me furious. Because aren't we, we're not just screwing up people, we're screwing up plants, we're screwing up water. And what about all the other species? What's happening to flora, fauna, little tiny things that we have no idea how important they are, not only to their own lives, but perhaps our well-being.
And I mean, I think your comment is probably how many people would react to a topic like this. And I have my own personal kind of approaches to it as well. In terms of contextualizing it, this is kind of why I brought this quote up here, because I really do think, and the way I was trying to explain it, is that scientists, when they start and they innovate and they make a compound, it's out of pure joy. It's the love of learning. It's about dissecting things apart. It's about taking those...
Organic chemist.
Yeah, so, and it's about knowing for the sake of knowing. And then what happens to that idea or that product or that synthesis after its initial kind of naive birth is where we have a problem, right? So someone can have a good idea and it gets passed off to someone who perhaps has more of a social agenda. I mean, when we talk about the great asymmetry, we talk about the guillotine, right? The physicists had figured out how to make this implement of destruction so that it was humane and just, and not just, well, I can't believe I said that. It was quick, okay? And guillotine worked on it really well. It was an engineering feat, but yet it was used in the hands of, you know, a less than pleasant cause. So I guess that's why I bring this up, is we're confronted with this great asymmetry, and Stephen Jay Gould is exactly imploring us to say, look, we have to think long term. You can't just, you know, it's not enough just to have this idea. As scientists, we have to guard our ideas. We have to make sure that they make it into the hands of people who will use them decently and ethically. And I don't think, I mean, there's no way to make that happen because, you know, there's always a profit to be made, you know? I mean, I think it's very germane to our political discussions right now, too. But I thank you for expressing that. Yeah.
Oh, I was just, maybe we started this. I just remember being, most of you would know, most of the people in this room would not remember this, but I remember growing up and in elementary school, regularly having these films that were showing about going into the third world and using all kinds of how modern Western chemical things were going to make life better for everybody in the world. And I think that's one of the things we have to always keep in mind is that a lot of these things were entered into with very strong, very positive motives. The problem that we always run into is, as you were saying, how does one gauge the long-term effects? My father died as a result of exposure to radiation of the Bikini atomic bomb tests. He was at the atomic bomb test in 1946. He died in 1998. And it took that long for the side effects to make themselves manifest. So I think we always have to be careful. On one hand,
Well,
it's understandable that we get worried about these things. At the same time, we were talking in court today about using mercury in medicine. Yeah, it works, right?
Right, but long term.
Long term.
Yeah, and that's what complicates the discussion even further, is that there are acute effects, there are intermediate latency effects, there are long-term effects, and you know, then we just get older, right? And so which of those effects are from old age, which are accelerated because of an exposure? And I mean, I think that's why we're still debating it. That's why we're still, hopefully, you know, more technology focused on this question in the appropriate way might yield some new insight. Anyway, anybody else?
If I could go back to the science, I guess I got a little confused about the auxins.
Yeah.
Because what I understood was that they were, this is how I understand it, at the edge of the leaves, and therefore that they would signal growth at a particular time.
Yeah.
But then you said that they discovered that, and so they thought, well, let's just use lots of auxins.
Right, because then it'll grow more.
And That's where I'm
but
losing it.
But instead of causing an appropriate amount of growth that could be maintained by the plant, it was too much growth at once,
Okay.
and it basically causes them to lean over and die.
Thank you, that's
So,
what I missed,
Yeah, yeah.
It affects root growth, doesn't it?
So auxins affect root growth, but in terms of doing that, it supports the material above the ground.
I have just a question on the chemistry part of it. Why chlorine? Because I noticed, the natural form of auxins, you've got, it's basically a hydrocarbon with nitrogen in there. But then I'm looking at all these synthetics, and the noteworthy molecules in there are carbon.
Yeah. Not all of them.
I mean chlorine. So why is it?
Not all of them are of that class.
Okay.
And that's about as best I can do on that question. It would have been, you make it, you test it to see if it has the effect, and then you just see what it is. So there must be something important about the shape of those molecules with respect to the chlorine, and of course, chlorine is electronegative, and so it may have some interesting interactions there. But I think that's as far as I can go on the chemistry, because I don't know.
Question. You're talking about the toxic effects and the long-term effects on the individuals exposed.
Right.
But isn't one of the greatest problems that's now starting to show up is the effect that the endocrine mimicry has on the next and the next and the next generation?
So I think what you're referring to is how dioxins mimic estrogens, and that of course that kind of exposure can alter sexual development, and so yeah. So the field, and again, I'm not a specialist here, but I have a little bit of an awareness. The idea of environmental estrogens is still pretty recent, and again, corpus of data is always kind of a problem in terms of coming up with a clear connection. But I do know that this is something that is now being collected in as a long-term effect that's being monitored by the Institute of Medicine, the IOM.
And particularly in Vietnam, where these photographs in the exhibit are of young children now.
So, in the field of epidemiology, I imagine there's a lot of statistics a lot of number crunching that has to go on before submitting your information. How, as people, can we, I guess, address the issue of some certain symptoms being statistically insignificant?
Yeah. So one of the things on the list from Sir Bradford Hill on the top was strength of association, and most of the work that's done after data are collected from subjects, be they from interviews, be they from hospital records, be they from actual taking blood or taking fat tissue, is to put all that together in a model, a statistical model that calculates a risk ratio that is governed by statistical rules. So there are conventions in terms of what that risk ratio or odds ratio has to be in order for it to be deemed a positive correlation. And so it's all governed by statistics. Yeah. But the point is you have to drill down in the data to where you're comparing apples to apples before you can make that statistical comparison for a difference between those populations. All right. Well, thanks again.